Hello guys, welcome to my channel. Today I will discussing about the Golgi apparatus. Okay, so let's start. This is the Golgi apparatus, a cellular organelles, a very important cellular organelles. Okay, so what is the situation? It is situated in between the endoplasmic reticulum and the cell membrane. It means it connects the endoplasmic reticulum with the cell membrane. Okay, so this structure is first discovered by a scientist known as Camillo Golgi. Hence, this structure is called Golgi apparatus or Golgi apparatus. Okay. So, we are seeing two phase. This is cis phase and this is trans phase. Why it is named as cis phase? It is named cis phase because of it is oriented towards the rough endoplasmic reticulum. It is oriented towards the rough endoplasmic reticulum, and the trans phase it is oriented towards the cell membrane. Okay, cis means towards the rough endoplasmic reticulum, trans means towards the cell membrane. Clear? Okay. So we are seeing this type of cellular structure, cellular folding. This type of structure is known as cisternae. Okay. This membranous folding is known as cisternae. There are 5 to 8 folding, means 5 to 8 cisternae in one Golgi apparatus. Means the number of cisternae can be in between 5 to 8. Okay. So this is the basic structure covered. Now we see what is the function. You know, the main function of rough endoplasmic reticulum. Rough endoplasmic reticulum means it is attached with the ribosomes. Ribosomes synthesize protein, and the protein is further processed in the rough endoplasmic reticulum. Some amount of partial modification, and then it is packed as reticular vesicle. Okay, it is packed at reticular vesicle and entered the Golgi apparatus through the cis phase. You seeing these reticular vesicles, okay, these blue colored reticular vesicles, these are entering the Golgi apparatus. Clear? So after the entering of the Golgi apparatus through the cis phase, the reticular vesicles, in the reticular vesicles there are some proteins, majority of the proteins, some amount of fat and some amount of carbohydrate and other molecules, etc., etc., mainly protein. Okay, because ribosomes are synthesized mainly protein. They in smooth endoplasmic reticulum there may be some synthesis of cholesterol, lipids, etc. So they are also present here. Okay. So after the entering of the Golgi apparatus through the cis phase, the proteins inside the reticular vesicles are released inside the Golgi apparatus, and they are further modified. Modified means they are folded here. They are associated together with some amount of carbohydrates, some amount of lipids, and because we know the phospholipids, glycolipids, lipoproteins. Okay, this type of modification occurs here, and then the modified products are packed as secretory vesicles. Okay, the products are packed as secretory vesicles. Reticular vesicles so converted into secretory vesicles. Secretory vesicles means there are some type of materials inside these vesicles which can be secreted. Okay, in outside the cell membrane or inside the cytoplasm. Okay, means secretory vesicles contains of secretory products. Okay, secretory products means it will be enzymes, etc., etc. Okay, so we completed the modification, we completed the packaging as secretory vesicles. Now what? There will be a labeling. Okay, labeling means in the substances which inside the reticular vesicles there must be some type of sequence, means a specific polypeptide chain contain a specific sequence of amino acids. Okay, so the on the basis or on the depending on the sequence of the amino acids or some or other compounds, the sequence have a specific information that where it to be supplied. Okay, where it to be supplied means if the protein is supplied to the mitochondria, then a specific sequence which is known as mitochondria localized sequence. Okay, there is a sequence known as mitochondria localized sequence. There are some type of other sequence. Okay, so they are labeled. Okay, secretory vesicles are labeled, that these vesicles will be transferred to the mitochondria, these vesicles will transfer to the other structure. Okay. Some of the secretory vesicles are also known as lysosome. We know lysosome, we will discuss later the lysosome. The lysosome is also a secretory vesicles. Okay, but it has a function, specific function. We will discuss it. Okay, so after the labeling, they are delivered to their respective appropriate structure. Okay, this is the function of Golgi apparatus.